So thank you to the organizers for inviting me to speak today. The title, the talk that I gave was actually the title of one of the two manuscripts I'm going to describe today, which is part of a project that we call ImmuneCODE, which is really around identifying T-cell response signatures to COVID-19. All the data I'm going to talk about today is available at immunerace.adaptivebiotech.com/data, as are the preprints. So first, some context. I presented at the AIRR-C meeting in Italy last year an overview of this project, but about 2.5 years ago, we launched this partnership between Adaptive and Microsoft that we call the TCR Antigen Map Project. And the goal of this project is really to develop the data and computational tools that allow us to map T-cell receptors to antigens to diagnose and treat disease. Just by way of brief background on the diagnosis side, in the last year or so, we've talked about a couple different examples of where we can really separate out cases from controls using repertoire sequencing. So one example I presented in the meeting last year had to do with imputing HLA types from repertoires. We can do that with well over 100 HLA alleles at this point. We've also talked about new methods that allow us to go back to the original data that started this project from Emerson et al. And improve on those results for classifying CMV infection. And then within the last year, we've talked about some new results in Lyme disease, where what we're seeing there is that public T-cell receptors are not only commonly found among individuals that are seropositive for Lyme disease, but among a sizable fraction of individuals who are clinically diagnosed with Lyme but are not seropositive. And we've now, this last summer, have launched a clinical trial to evaluate the clinical utility of a T-cell receptor-based diagnosis. So, of course, when COVID came up last winter, the obvious question was, what can the Antigen Map project do for COVID? So starting around February, we started looking into this, and in March, we launched this new project that we call ImmuneCODE. And at a high level, the goal was really to get as much data as we could and release it to the community. There's a couple sources of data. I'll talk about those in the upcoming slides, but broadly, there's a sponsored study that Adaptive is running for ongoing enrollment of patients. And then we've really leaned heavily into developing partnerships with various groups around the world who have blood from COVID patients, and then we've been doing the TCR sequencing. The other two types of data I'll talk about more include mapping from T-cell receptors to actual antigens, which one of the earlier speakers discussed today as well. And then we're also doing a lot of repertoire sequencing of patients directly. So let me spend a few minutes and describe the data. Again, this is all available at the website. The first type of data that was discussed earlier comes from the MIRA assay that Adaptive developed, which is really about mapping T-cell receptors to antigens. So I'm going to spend a few minutes and just walk you through the data sets that are available here, and I'll point out where you can go and find these. So right now, the data that we have publicly available are HLA class 1 restricted peptides mapped to CD8 sorted T cells. And those constitute 545 peptides that were chosen last winter, early spring, based off both NanoIMHC predictions, but also published peptides. These span the entire proteome and have been binned into 269 unique addresses, where the binning was really based off overlapping peptides where we expected there to be high degrees of cross-reactivity. So details from that are in one of the two papers that I noted before. All of these in these gray boxes just point you to how you can pull the numbers that I'm sharing in the slides, so that you can navigate that yourself. One of the new things in this MIRA assay that we've released in this data set is transfection experiments. So the classic MIRA experiment uses peptides that are exogenously used to stimulate T cells. The problem with that, of course, is that if you put peptides into the experiment that are not naturally processed and presented, you can still get results, even though those would not be physiologically relevant. So there's a version of this assay that uses DNA mini-genes that are transfected into antigen-presenting cells. And we can use that to then validate the actual processing and presentation. The design of this experiment was to include mini-genes that cover the entire genome with the exception of ORF1ab, because it's so big. And there's a handful of mini-gene experiments that are currently available on the website. Thus far, we have HLA data available for 84 donors, so that will be a rich resource as well. And then there's a few different experiment types. So I just wanted to describe those a little bit because they're mixed together, and I just wanted to be clear what types of data are available and how you might use them. So one of the types I think of as the effector memory T cells. These are not actually sorted for effector memory T cells, but is more based off PBMCs and using the fact that those cells that are already expanded are going to be predominantly effector memory T cells. And you can pull those by looking for the PBMC cell type in that data set. Within that group, we have done MIRA experiments for 4 active infections, 58 convalescent patients, and currently 1 exposed uninfected patient. So most of the data that you'll pull out of that is actually from convalescent patients. Again, you can pull that from the metadata file. There's also currently 5 healthy donors that do not have any known exposure history, although of course if these are recently pulled samples, it's hard to know that for sure. And then there's another set of experiments that are naive T cells. These are cells that are actively sorted for naive markers and were pulled from healthy donors. And currently there's 27 experiments in that data set. So the naive T cells and the effector memory T cells have different use cases. Everything I'm going to show you for the rest of the talk is going to focus on the effector memory responses. These are actual responses we're seeing in patients. The naive T cells are really a good way to get a better understanding of the theoretical landscape, but they don't measure, of course, what is actually happening in patients. And thus far in the current release, there's about 184,000 T cell receptor antigen mappings. I will say that Adaptive has started generating HLA class 2 restricted responses as well. This is, of course, a different assay to get this to work. That's an overlapping peptide design, and we expect that data to be available in the next release. The other type of data that we are generating is really focused on repertoires, and the goal here is to identify shared TCR signatures across populations. In the current release, there's about 1,400 samples from 1,200 individuals, about 350 million unique TCR beta sequences. These are all bulk sequencing. The data come from really 2 major sources. The first is from partner cohorts. Here's a list of the cohorts that are currently available. There's more that will be coming. I did want to highlight one of these cohorts that will be interesting to this community, and this is from ISB, the Institute of Systems Biology in Seattle. If you're not familiar with the study, I recommend you go to this website. In addition to the T cell receptor sequencing, they're doing a lot of genetic profiling, they're doing proteomics, they're doing microbiome, of course, HLA types, serology, et cetera. So this will be an interesting resource for the community and a really nice way to pair repertoire data, TCR repertoire data, with other modalities. And then finally, the Adaptive has launched an ImmuneRace study, a study they call ImmuneRace, where basically anyone in the US, we are limited to the US right now, can enroll at the website. And those that are qualify, then LabCorp's Covance unit will send out mobile phlebotomy to your home to actually draw blood. So this is a good way to specifically get different targeted populations. And that's an ongoing study and an ability for us to target sample collection. All right, I'm going to spend the next few minutes and talk about some pretty high-level descriptive results from what we've seen in these data. The first is looking at antigen specificity, and the question here is really where are CD8 T cell responses focused? So the next series of slides are all going to be figures from the preprint, the Snyder et al. Preprint that's available on medRxiv, so you can go read more about this, but I thought I'd just walk through these results at a high level. So the first result I want to show is just the total breadth of the responses. And just to orient you to this figure, the gray bars are where we actually had peptides, so we could actually look for responses. And the blue lines on the top are the total number of unique TCRs that have been identified across the experiments on a log scale, and the number of unique individuals that had at least 1 TCR mapping to that antigen across the 61 or so experiments that were done at this time. So at a high level, you can see that we see responses across the entire proteome. There's no particularly obvious spikes by or immunogenic regions at this scale. As you'd expect, the vast majority of responses are not observed in a majority of individuals because these are HLA class 1 restricted. And when you do see spikes, these tend to be in adjacent regions, not the same address space. I mean that those are different epitopes being presented. One way we can look at this then is to look for immunodominance. So as I said, there are no antigens for which a majority of people are responding, but that's because of HLA restriction. If you limit by HLA restriction, we identify a number of epitopes that are very commonly targeted, a number of these epitopes that we observe responses in 100% of individuals with the presenting alleles. Perhaps the most studied across the community is this AO101 restricted HY11 epitope in ORF1AB. We and others have noticed that this is targeted by essentially everyone that has AO101, which is pretty interesting. It's 100% of our donors that are targeting that. One thing you can do with these response data is cluster individuals, cluster the donors by the actual antigens that they're targeting. The thing that jumps out immediately is a handful of these clusters that are, of course, HLA restricted. This one on the upper left is the HY11 epitope. This other one is a B0702 restricted epitope. And you can, of course, then also collapse that down to proteins, which largely reflects the epitope level immunodominant structure. Now, at this point, we treated this as a largely descriptive exercise, though, of course, there's a lot of really interesting questions that come up that I think are very important for the community to look into and that we're starting to look into ourselves. The first is whether antigen specificity correlates with outcomes. There's lots of ways to look at the role of T cells with outcomes, but I think one interesting question that these data allow us to start probing at is whether the specificity of the antigens makes any difference. Another question is whether pre-existing immunity will actually re-architect these immunodominance hierarchies. And we don't really have the data to poke at this yet, but I think this will be a really critical question as vaccine study data, for example, starts becoming available. If you say, induce a response only against spike in this one protein, will you actually reorient the immune responses of these AO101 individuals that would otherwise predominantly target ORF1AB? Those will be interesting questions. Of course, the other way you can get pre-existing immunity is through cross-reactivity to endemic coronaviruses. On the shared T cell receptor side, the question here is whether we can identify signatures, really public T cell receptor clones that associate with COVID-19 disease across these different cohorts that span really Europe and North America at this point. So perhaps the simplest way we can look at this is with these enhanced sequence plots. And all we're doing here is first identifying with Fisher's exact test public clones that associate with the endpoint, in this case, COVID-19 diagnostic. And then on the y-axis, we're showing here the number of these COVID-19 associated TCR public clones versus the number of unique clones that were pulled out of the repertoire. In this particular case, we trained the model, identified these enhanced sequences on 2 cohorts from Italy and New York. And we see these are really nicely generalized to 3 completely independent cohorts. Overall, we're identifying about 4,000, a little more than 4,000 public enhanced sequences that really define this signature. I'll note here that the controls in these cases were about a little over 3,000 historic controls from pre-2020 that we split evenly between training tests. Now, there's a subtle point here that your eye might pick up, which is that these orange dots are all shifted to the left. Some of this is just assay differences, sequencing depths, sample quality issues. But of course, you can also look directly at the T cell fractions that are in blood and PBMCs. And by and large, we see what you'd expect with COVID, which is that the recovered individuals have high T cell fractions that are comparable with what we see, what would be a typical T cell fraction. Whereas all these other cohorts that are mostly acute patients have much lower T cell fractions with a pretty sizable subset that are extremely low T cell fractions. There's of course some interesting biology going on that also raises some challenges from an assay or computational standpoint when you try to look at these things. Now, there's many ways we can look at these public clones. 2 measures that we like to look at that are very simple. One is clonal breadth, which we define as really just the normalized number of distinct SARS-CoV-2 specific or associated clonal lineages. So just the number of enhanced sequences normalized by the total number of unique clones pulled out of the assay. And the other is depth, which is related to the total expansion of those lineages. And by either of these methods, what you see very quickly is that there's a rapid expansion of these SARS-CoV-2 specific T-cell receptors that expand over time and persist late into infection. If we zoom in on the holdout test set and just think about breadth here for a moment, you can imagine drawing a line here that essentially defines a threshold above which we've ever seen a SARS-CoV-2 breadth in historic controls, and ask how common are these actual shared clones among COVID patients? And the answer roughly is that early in infection, roughly 75% of these individuals have a very high breadth of these public T-cell receptor clones, and later of infection, that proportion goes even higher. In the paper, you can see how we break it down in more detail. But overall, we're seeing these expanded public clones persist for at least a couple months since the clearance of symptoms. And of course, as this epidemic goes on longer, we're able to get data from farther and farther out. Now, of course, there's a number of questions we'll want to ask about this. Again, I think really important questions for the community to address, and we'll be trying to address as we get more data. The first is, do the false negatives have different TCR signatures or simply a lack of response? This was raised in one of the earlier talks as well. We simply don't know yet, and it may well be that the answer to this question differs whether you're looking early or late in infection, but I think that will be very important. And of course, the more general question is, do TCR signatures associate with outcomes? And perhaps even more critically, is there a causal link with outcomes? I don't have any data to present at this point. We're really trying to get more clinical outcomes and really start probing at this question. Another way we're starting to look at these data is, how do we actually identify antigen-specific clones in repertoires? So the ideal experimental design at a global pandemic level would be to do some sort of antigen-specific assay across thousands or tens of thousands or even millions of patients. But of course, none of these assays scale to that level. What we can do, though, is we can contemplate repertoire sequencing for many thousands of individuals. And so what can we actually infer from a repertoire without the actual functional mapping? So there are a couple of challenges to this, and the first is that antigen-specific clones are mostly private. There's many ways to look at this. People have seen this in many different ways. One very simple way to look at this that we explored briefly in the manuscript was, if you simply take the matched MIRA, or the MIRA from an individual, and then ask how many of those mapped TCRs show up in a repertoire sequencing run via the normal assay, you get about 25% overlap, which is roughly what you'd expect given the different levels of sensitivity of those assays. But if you take a library of matched T-cell receptor clones from other individuals, there's actually very little overlap, indicating that most of those responses we're picking up are actually private. Now, that said, about 10% of the enhanced sequences exactly match a MIRA mapped sequence. So by construction, the enhanced sequences are public clones, which are enriched in cases versus controls. And a small fraction, but an important fraction, actually can be mapped directly to antigens. And you know, I'm not going to go into too much detail here, but at a high level, we see that we're mapping at exact hits across the entire proteome. A sizable portion of these hits are going to this immunodominant AR1 epitope, but we're seeing it to a number of immunodominant epitopes as well. And in some sense, this isn't very surprising. We and others have described the very immunodominant epitopes in different HLA contexts, and it turns out there are some very public clones that are specific to each of those epitopes, which allow us to start picking up these signatures. And finally, at kind of a macro level, you can look at the breadth and depth of the public enhanced sequence response, and it turns out that that correlates quite nicely with the private breadth and depth you can measure directly in these patients. And again, these are early data. I don't want to read too much into those, but this is another figure from the manuscript that simply showed that if you describe the overall breadth of the response via directly mapping to those 550 or so antigens, and compare that with the breadth that you would estimate from public clones, it actually correlates pretty nicely with some notable outliers. We would expect this. We're only looking at CD8 responses. Certainly others have shown that CD4 responses are critical and quite possibly the majority of the responses. But nevertheless, we see a really nice correlation here, which is simply to say that we can infer a shocking amount of detail about the overall breadth and depth and even the antigen specificity just by looking at these very public clones. So with that, I'm going to go ahead and wrap up. I'm happy to take more questions. At a high level, you know, the first thing we sought out to do was to determine whether there was actual distinctive TCR signatures in COVID patients. We have in fact seen that. Adaptive announced a month or so ago that they are pursuing an emergency use authorization for a diagnostic around this. So I think that's interesting clinically, but also pretty exciting from the standpoint of actually getting a TCR-based diagnostic into the clinic and trying to understand what the clinical utility of that will be. We are, of course, continue to get more data. We'll have more repertoires from more cohorts from more geographical regions coming soon. We're really focused on trying to get more endpoints. There's actually a lot of endpoint data in the public release right now, but we're working on getting more clinical endpoints as well. And as I mentioned earlier, we'll be releasing CD4 data soon, which should help clear out some of that picture. And some of the key questions as we continue to look forward really come down to outcomes. Do TCR signatures correlate with outcomes? Does the antigen specificity correlate with outcomes? Are either causal? I didn't talk about that at all today, but we do have longitudinal samples for maybe 100 or so cases and are looking to fill that out both with more time points and more individuals. I haven't really wanted to dive into that too much yet until we can really get a richer data set, but I think that will be a really interesting opportunity to look closer at causality. And really what we all ultimately care about is what constitutes protective immunity, which of course is a difficult question to directly address. So with that, let me stop and I will refer back to the publications themselves for acknowledgements. There's a lot of folks both within Adaptive and Microsoft, but also a number of our clinical partners that contributed data that I want to be sure to thank.
Great, thank you very much. Okay, so the first question is how do the HLA class 1 restricted peptides link to other antigenic specificities? For example, are some of these peptides also found in IEDB, for example?
Yeah, no, that's a great question. I kind of glossed over that. So the panel selection was done back, I don't know, March or so, and there were two criteria. One was published epitopes, so that came out of IEDB, and then the other one was NetMHC predictions for common HLAs. One way to go back and look at that is actually to ask, okay, for the peptides we have, how conserved are those peptides, either phylogenetically across all coronaviridae or specifically across the common, the four so endemic coronaviruses? I'd say, depending on how you count, roughly 10% of those peptides are very well conserved with the endemic coronaviruses. Mm. That'll certainly be an interesting place to dig into the role of pre-existing immunity in terms of the cross-reactivity and potentially linking that with outcomes.
Mm-hmm. Great, thank you. So another question, I was trying to reconcile the fact that if you can show the slide where you have the viral genome along the x-axis, and then along the y-axis, it was showing, I think, the number of T cells you pulled down that were responding specifically to that peptide. And you indicated here that you find basically no cross-individual commonality in terms of finding a T cell that's responding to exactly the same peptide. Did I understand?
Oh, no, I'm sorry. What I meant was there were no, as expected, there's no epitopes for which the majority of individuals are targeting that epitope. And this really comes down to HLA restriction. These are class 1 restricted peptides, and there are, in most populations, no class 1 alleles that are in a majority of individuals.
Right.
So if you restrict by HLA type, then you very much get these highly immunodominant epitopes where 100% of individuals with AO101 are responding to this HY11 epitope, for example. But it's just that we don't.
Right. Okay. So then my question is, So my question then is, how does that relate to your finding of what looked like a substantial number of public clones? Is the clone sharing then only across individuals who have the same HLA allele? Or how would you
There's
tell
two ways
us
you can
about
do that. I kind of glossed over that. You could certainly identify public clones at a per allele level. At some level, I'll tell you that once we have 1,000 cases and many thousand controls, that kind of averages out, right? If you identify public clones that are specific to this HY11 epitope in AO101, depending on the cohort, 10 to 30% of individuals have AO101. So now if you pool that with thousands of public clones that are specific to other HLAs, that actually gives a very good separation. You can certainly go in and you can separate that out, and you can imagine more clever ways to be HLA specific. But at an aggregate level, you can do really quite well. So if I skip ahead to say this plot here, this is all these plots on the left are doing is summing up the normalized number of 4,000 public COVID-associated clones in cases versus controls. And like I said, you can very easily go back in there and map most of those individual clones to individual HLAs where we have HLA types, and we're certainly doing that. But at a macro level, once you average over those 4,000 clones, you really don't need to do that to get good separation. That said, you have to be very careful about that. You have to be sure your cases and controls are well balanced by HLAs. Again, I didn't show you any of that data. That's something we always do. One of the most important use cases for imputing HLAs from repertoires, I think, is to make sure that your cases and controls are well balanced, and to fix any imbalances that you have, and to explain away confounding that's due to HLA. So absolutely, that's an important step. I don't want to gloss over that.
Right. Well, and then another question, and maybe you answered this in your answer, and my apologies, I was getting an unstable internet connection message from Zoom during part of your answer. But do you think if you looked at particular motifs rather than full-length sequence, that you would find more public responses? So doing something like what the glyph algorithm does, for example.
Yeah, absolutely. We're certainly doing that with both available and algorithms that we're developing ourselves. I think that will certainly be an important aspect to this. Now, there is a specificity issue with any machine learning problem when it comes to identifying these motifs, which is that if you then go to apply it to repertoires, you need to essentially have 100% specificity to not be overwhelmed by false positives. And it depends on the application. So what I presented here was exact matches because we can be very confident in the specificity there, but it is certainly the case that we want to look at signatures. We just have to do that in a very careful way so that we're not overwhelmed by those false positives and some of the confounding that can come with that.
Mm-hmm. Okay, great. And then there's two last questions here in the chat, which I'll try to squeak in. I think we have 1 minute left. Do you think that some of the public clones bind to multiple viral antigens, not just to SARS-CoV-2?
It's an interesting question. Certainly the endemic coronaviruses, I would absolutely expect that to happen. The cross-reactivity otherwise, I don't know how conserved these are with other viruses. That would be an easy thing to look up with the tables that we publish. My sense is that that's not very likely, but it would be a good thing to dig into and better understand.
Mm-hmm. And do you see any interesting repertoire features that associate with particular viral antigens, such as nucleocapsid versus a receptor binding domain, etc.?
In terms of like breadth and depth, sort of those, or clonality, those sort of.
Yeah, I mean, I think it's an open-ended question in terms of which type of repertoire feature you may have noticed being associated.
Yeah, we haven't looked in a ton of detail on that. I think that's a really interesting question, and that will also be wrapped up even more so with HLAs. So, that HY11 epitope is dominant, not just in the sense that that most people, if not everyone, with AO101 and SARS-CoV-2 target it, but it very much dominates the response. And how much does that actually suppress other responses? I don't know the answer to that. Does that mean that AO101 individuals will have different overall clonality because they're so focused on that one epitope? I think those are all really interesting questions that need to be explored. And then again, if you vaccinate an AL101 individual against spike, which doesn't include that epitope, how does that re-architect the immunodominance hierarchy? Will be, of course, a critical question as vaccines come online.
Okay, great. Thank you. So with that, it's time for us to transition now to our moderated panel.